The purpose of this project was to determine whether mycorrhizal powder increases the growth of legumes. Arbuscular mycorrhizal fungi, or AMF, are a fungus that grow in a symbiotic relationship with the roots of plants. The plant offers the fungus a place to live in nodules on the root, along with nutrients from photosynthesis, and the fungus aids in nitrogen fixation and uptake of other nutrients from the soil. The legumes used in this study were Phaseolus lunatus, or lima beans. According to an analysis of various AMF products done by Suleiman Basiro, AMF have become increasingly popular and effective in agriculture after being proven to improve crop productivity. AMF products were proven effective in this study in nutrient uptake, plant growth induction, and climate stress alleviation. Based on Basiro's study and other similar research, we hypothesized that mycorrhizal fungus influences plant growth. This led us to predict that in the presence of mycorrhizal powder, the growth of the lima beans would increase. We potted 30 beans with a half tablespoon of mycorrhizal powder in topsoil and 30 beans with just topsoil. All beans were stored in the greenhouse at a constant temperature, and the seeds were watered with 20 milliliters every 3 days with water from the greenhouse sink. The amount of water, controlled environment, same pot type, and same type of soil acted as our constants. Once we measured for 3 weeks, we then dried them and measured their weighted biomass and the length of their roots. There are some possible errors that could have impacted our results. One error is that not all the plants germinated. Another is that the temperature could have varied in the greenhouse based on the time of day, thus not keeping it constant for growth. The last error was that the greenhouse has sprinklers on the sides that could have activated at any moment, which may have disrupted our watering patterns and schedule. The graphs show that there was a variance in the different groups across the weeks. The beans with the added mycorrhizal powder had the higher average height until week 3, when the beans without the added powder had the higher average. The same was shown for leaf growth over the 3 weeks. This could be due to some unknown factor that impacted the growth of the beans or limited the function of the mycorrhizae. Another interesting data point we found was that the average biomass was higher for the beans without the mycorrhizae powder, yet the average root length was higher for beans with the mycorrhizae powder. This can be explained by a study found in the Fields Crop Research Journal stating that mycorrhizal relationships can increase root surface area for nutrient absorption. Based on literature, mycorrhizal powder has become a popular inoculant to improve crop productivity. The results we gathered regarding plant growth in our 3 weeks of testing were not significant enough, however, to draw any conclusions, and thus did not support our hypothesis that mycorrhizae influence growth. However, our finding of an increased root length aligns with the literature that mycorrhizae can influence the surface area of the roots. This is a significant finding because it allows plants to absorb more nutrients through the soil and thus leads to more productive plants and crops.